My name is Olivia Piacentini. It is June 13th, 2024. Pause to read case 3. The information suggests that the patient likely has type 1 diabetes. Insulin is a hormone secreted by the pancreas that is involved in glucose homeostasis through promoting glucose transport across plasma membranes of targeted cells. Activation of this process occurs due to a ligand-induced dimerization that causes a cascade of phosphorylation and dephosphorylation events. Insulin will bind to extracellular alpha subunits, which causes an autophosphorylation event to occur through the beta subunit on tyrosine residues within the cell. This activates insulin receptor substrates to recruit other proteins. Two important proteins are GRUB2, which will activate a MAP kinase cascade, and IP3, inositol triphosphate 3 kinase, which can activate the downstream kinase PDK1. This protein phosphorylation causes the glucose transporters, GLUT4, to be embedded in the cellular membrane. This allows glucose to be transported in the cell for processes such as glycogen synthesis, fatty acid synthesis, cholesterol synthesis, and protein synthesis to occur. The uptake of glucose to be used for the previously stated events to lower blood glucose levels, returning an individual to homeostasis. The low serum insulin levels from the patient indicates dysfunction in production of insulin in the pancreas, likely associated with the condition type 1 diabetes. Given the patient's condition, I would predict that the patient's fasting glucose levels would be above the normal range. As previously outlined through the explanation of the insulin signaling pathway, insulin is necessary for the transport of glucose into cells. This transport allows for a variety of processes such as glycogen synthesis, fatty acid synthesis, cholesterol synthesis, and protein synthesis to occur. Removal of glucose from the bloodstream into the cells allows for these processes to occur while returning the blood glucose levels back to homeostasis. However, as the patient has low serum insulin levels, his body will not be able to trigger the cascade of events since there will be no insulin to bind to the cells, allowing the body to utilize glucose in the blood for necessary biological functions. Two likely regulatory enzymes active in this individual's condition are hormone-sensitive lipase and glucose-6-phosphatase. Hormone-sensitive lipase is an important enzyme in the lipid metabolism. It mediates triglyceride hydrolysis to free stored fatty acids and glycerol through the process of lipolysis. These products are then oxidized in liver and skeletal muscle cells. In an individual without diabetes, insulin dephosphorylates and inactivates lipolysis. This prevents the accumulation of free fatty acids. However, since the individual has low levels of insulin, lipolysis in hormone-sensitive lipase is unable to be inhibited and therefore remains active. This causes an accumulation of free fatty acids, which can cause many issues with homeostasis throughout the entire body. Glucose-6-phosphatase is an important enzyme in gluconeogenesis and glycogenolysis. It is responsible for phosphorylating glucose-6-phosphate into free glucose to be released into the bloodstream. In normal individuals, when blood glucose levels are high, insulin will inhibit glucose-6-phosphatase in order to lower blood glucose levels to regain homeostasis. However, since the patient does not have adequate levels of insulin, his body is unable to inactivate glucose-6-phosphatase. Since glucose-6-phosphatase is active in the patient, causing blood glucose levels to rise, and his body is unable to use insulin to remove glucose, he will become more hyperglycemic. Pause to read case 4. Hereditary fructose intolerance is caused by a disorder in the fructose metabolism through a deficiency in aldolase B. To understand this disorder, it is important to first understand fructose metabolism. Fructose is a major dietary component in sucrose. It is primarily metabolized in the liver and is taken up by cells through facilitated diffusion using GLUT5 transporter. The initial steps of metabolizing fructose is through the phosphorylation to fructose 1-phosphate. Fructokinase is the major enzyme responsible and rapidly phosphorylates fructose as it enters into the cell. Next, aldolase B will cleave fructose 1-phosphate into dihydroxyacetone phosphate and glyceraldehyde, which can enter the glycolytic pathway as glyceraldehyde 3-phosphate. Aldolase B is the rate-limiting step for the fructose metabolism. It has a much lower affinity for fructose 1-phosphate than it does for fructose 1,6-bisphosphate. Therefore, after consuming a high dose of fructose, normal individuals will accumulate levels of fructose 1-phosphate and slowly convert it into glycolytic intermediates. As aldolase B is the rate-limiting step in the fructose metabolism and is responsible for breaking down fructose 1-phosphate into glycolytic intermediates, hereditary fructose intolerance results in the accumulation of fructose 1-phosphate. The accumulation of fructose 1-phosphate will eventually inhibit both the process of glycogenolysis and gluconeogenesis due to the lack of free phosphate. Fasting metabolic pathways require phosphorylation of the regulatory enzyme to allow for activity. The reduced levels of free phosphate will impair the ability to generate ATP in the electron transport chain. Clinically relevant symptoms can include hepatomegaly or enlarged liver, fasting hypoglycemia, and irritability after eating fructose. Relating back to the patient, the mother reports the infant has only ever eaten breast milk. Breast milk is primarily composed of lactose, which is glucose and galactose. Therefore, it is unlikely the patient is affected by hereditary fructose intolerance, as this condition is caused by a disorder in the fructose metabolism and usually revealed by the introduction of fructose-containing food in the child's diet. Galactosemia is characterized by a deficiency in the key enzyme within the galactose metabolism, galactose 1-phosphate uridyl transferase, or GALT. Again, it is important to understand the proper functioning of the galactose metabolism first. Galactose is ingested as a disaccharide of galactose and glucose called lactose. Disaccharide lactose is ingested in the intestine to its monomeric sugar, glucose, and galactose. Galactose is taken up by the cell and is phosphorylated into galactose 1-phosphate by the enzyme glucokinase. Galactose 1-phosphate is then activated into the uridine diphosphate sugar, UDP, through GALT. The metabolic conversion can use UDP glucose for glycogen synthesis or can convert it into glucose 1-phosphate. This can then be converted back to glucose 6-phosphate to enter the glycolytic pathway. Galactose can also be converted into glucose 1-phosphate, become an active sugar, and then be used for glycogen synthesis, with the use of galactose 1-phosphate uridyl transferase. It can also be used for synthesis of lactose in milk production. In galactosemia, affected individuals are born with a deficiency in GALT. This deficiency will cause toxic levels of galactose 1-phosphate in the liver. This inhibits hepatic glycogen metabolism and other pathways that require UDP sugars. Furthermore, this accumulation of galactose in the blood will then cause the body to reduce it into galactitol, its sugar alcohol form. Galactitol can cause damage to many different tissue types. Clinically, this condition can manifest through feeding intolerance, hepatomegaly, lethargy, renal disorder, and clotting impairments. Relating back to the patient, as reported by the mother, the primary food source is breast milk. As we know, breast milk is composed of lactose, which is glucose and galactose. However, if the patient was affected by galactosemia, it would be more likely that her symptoms would have arisen more quickly, as individuals who are affected will present in the first few days of life. Since the patient has only had breast milk, which contains galactose and glucose, it is unlikely she would have been asymptomatic and healthy for 2 months prior to showing symptoms. Glucose 6-phosphate dehydrogenase is a key enzyme in the pentose phosphate pathway, which is a cytosolic pathway. The pathway does not produce ATP directly through the oxidation of glucose 6-phosphate. This pathway is important for the production of NADH, ribose 5-phosphate, a 5-carbon sugar used for DNA nucleotide synthesis, and substrates for glycolysis, fructose 6-phosphate or glyceraldehyde 3-phosphate. There are 2 parts to this pathway, the oxidative phase and the non-oxidative phase. During the oxidative phase, 2 irreversible oxidations that produce NADPH occur. This NADPH is used for reductive, detoxification, and fatty acid synthesis. The non-oxidative phase allows for the conversion of ribulose 5-phosphate into ribose 5-phosphate, and it can function independently when demand is high. Conversions through enzymes transketolase and transaldolase are reversible. These enzymes will move 2 or 3 carbon units onto other sugars to generate various sugar intermediates to create glyceraldehyde 3-phosphate or fructose 6-phosphate. Glucose 6-phosphate dehydrogenase is a regulatory enzyme within the pentose phosphate pathway. It will oxidize glucose 6-phosphate to 6-phosphogluconolactone, which will then undergo further oxidation and decarboxylation to result in ribulose 5-phosphate. Glucose 6-phosphate dehydrogenase is regulated negatively by NADPH. High levels of NADPH will inhibit its activity, so NADPH is only generated when needed. Glucose 6-phosphate dehydrogenase deficiency can cause acute hemolytic anemia caused by the destruction of red blood cells. It is a common disorder caused by a sex-linked mutation. Glucose 6-phosphate dehydrogenase protects red blood cells from harmful reactive oxygen species, which are byproducts of cellular metabolism. A deficiency in glucose 6-phosphate dehydrogenase will reduce its ability to protect these red blood cells, which will result in damage to them. This condition is unlikely to be affecting the patient, as common triggers of symptoms of hemolytic anemia include ingestion of fava beans, certain drugs, or an infection. There were also no remarkable symptoms that the patient presented with that would support a diagnosis of glucose 6-phosphate dehydrogenase deficiency. To understand glycogen storage disease, it is important to know how glycogen is made and broken down. Glycogenolysis and glycogen synthesis are essential pathways for glucose homeostasis through supplying and supporting blood glucose levels. When insulin levels are high in the fed state, glycogen synthesis occurs. Glucose is taken up and phosphorylated to glucose 6-phosphate. It is then isomerized into glucose 1-phosphate, which, in addition to UTP, is accepted by the enzyme UDP glucose pyrophosphorylase to generate UDP glucose. UDP glucose is a source for glycosyl residues to link to the glycogen chain. A regulatory enzyme for glycogen synthesis is glycogen synthase, which is responsible for linking the glucose monomers to the glycogen polymer. Also, when insulin is high in the fed state, the activity of cyclic AMP-dependent kinase is reduced, allowing for dephosphorylated glycogen synthase to remain active and glycogen phosphorylase to be dephosphorylated and inactive. During glycogenolysis, where the breaking down of glycogen occurs, the enzyme glycogen phosphorylase will free glucose 1-phosphate from stored glycogen. Free glucose 1-phosphate will then be isomerized to glucose 6-phosphate. In the liver, the glucose 6-phosphate will be dephosphorylated by the enzyme glucose 6-phosphatase. In the fasted state, either glucagon or epinephrine are elevated to stimulate the production of cyclic AMP. This will then activate glycogen phosphorylase and deactivate glycogen synthase. This process breaks down stored glucose, glycogen, in the liver to release into the body. The glucose is used in glucose homeostasis as well as a substrate for red blood cells in the brain. Glycogen storage disease is characterized by an individual's inability to break down glycogen into energy, causing a toxic accumulation. These individuals lack or have a mutation in the enzyme glucose 6-phosphatase. Glucose 6-phosphatase is present in the liver and is required for both gluconeogenesis and glycogenolysis. It is necessary to phosphorylate glucose 6-phosphate to free glucose to release into the bloodstream. This failure to maintain glucose homeostasis results in an increase in lactic acid production, because the accumulation of glucose 6-phosphate stimulates the glycolytic pathway. Clinically, deficiencies in glucose 6-phosphatase and therefore glycogen accumulation in the liver can lead to hypoglycemia, hepatomegaly, lactic acidemia, ketosis, and hyperlipidemia. Considering the pathophysiology of the disorder alongside the patient's presentation, the most likely diagnosis for the patient is a glycogen storage disorder. The hypoglycemia and hepatomegaly are classic characteristics of the disorder. The patient is unable to maintain glucose homeostasis as she enters into the fasted state, as she cannot break down glycogen into free glucose. And this accumulation of glycogen in the liver is what's causing hepatomegaly. Additionally, when epinephrine is administered, there is no increase in blood glucose, but there is an increase in blood lactate. Epinephrine should facilitate glycogenolysis in the liver. However, since the patient is missing the enzyme glucose 6-phosphatase, she is unable to phosphorylate glucose 6-phosphate into free glucose, causing hypoglycemia and lactic acidosis. However, there are many different types of glycogen storage disorders, and further testing would be necessary to determine the particular one that affects the patient.